Welcome dear students. Today we are going to discuss the Krebs cycle, also called as the citric acid cycle or tricarboxylic acid cycle. It was first introduced by Hans Adolf Krebs in 1937, and because of the contribution of this biochemist to the completion of the Krebs cycle, he was awarded with Nobel Prize in 1953. It is called as citric acid cycle because the first product of the Krebs cycle is citric acid, and citric acid is a tricarboxylic acid which contains three carboxyl functional groups. That is why it is also called as the tricarboxylic acid cycle or TCA cycle. My dear students, all the nutrients in our diet, they are either in the form of carbs, fats or proteins. So when we take our diet, that nutrients are first digested in the stomach and then they enter into the cells after the reabsorption, and carbs, fats and proteins through different metabolic pathways, they are changed to acetyl-CoA enzyme A. The carbohydrates, through some steps of the cellular respiration, they are converted to acetyl-CoA enzyme A. The fats, through beta oxidation, are converted to acetyl-CoA enzyme A, and proteins are first deaminated and then they are converted into the acetyl-coenzyme A. Now, this acetyl coenzyme A enters the Krebs cycle, which uses the acetyl coenzyme A, and it produces energy-rich molecules like adenosine triphosphate (ATP), reduced nicotinamide adenine dinucleotide (NADH), and reduced flavin adenine dinucleotide (FADH2). And these NADH and FADH2, they are energy-rich molecules. When they enter in the electron transport chain, then after oxidative phosphorylation, they also produce the ATPs. One NADH produces 3 ATPs and one FADH2 produces 2 ATPs. And my dear students, it means that the Krebs cycle is a metabolic cycle, metabolic pathway, which is used to change the nutrients into the chemical energy in the form of the ATPs. Now, my dear students, carbs, fats, and proteins, all these three, they are components of our diet and they are used to produce energy in our body. But the major contributor of the energy in our body, they are the carbohydrates. Now, the carbohydrates, first, they are digested to a very important monosaccharide, that is the glucose, and then glucose is used to produce the energy in the form of the ATP. And all those metabolic pathways which are used to completely oxidize glucose into carbon dioxide and water and to produce the ATPs from the glucose, they are collectively called as the cellular respiration. So cellular respiration simply means that all those reactions, all those metabolic pathways that completely oxidize glucose to the energy rich adenosine triphosphate. The first step of the cellular respiration, we have already discussed it in very detail in a separate video lecture, that is the glycolysis, in which one glucose molecule is converted to the two pyruvates. And my dear students, then this pyruvate, this enters the mitochondrial matrix because the glycolysis takes place in the cytoplasm of the cell. So pyruvate then from cytoplasm, it enters the mitochondrial matrix where it reacts with acetyl coenzyme A. Then it is decarboxylated, mean the carbon dioxide is removed from the pyruvate, and then my dear student, it is oxidized and the hydrogens removed are used to convert the NAD into the NADH. No, because the pyruvate is oxidized as well as decarboxylated, so this reaction is called as the oxidative decarboxylation, and as a result of this reaction, the pyruvate is changed to the acetyl coenzyme A, and this step, that is catalyzed by a group of enzymes which are called as the pyruvate dehydrogenase complex PDC. So that was the second step of the cellular respiration. Now, my dear students, the third step, it is the Krebs cycle that we are going to discuss today. And then the energy-rich molecules NADH and FADH2, which are produced in the Krebs cycle, they enter in the electron transport chain and oxidative phosphorylation, and they are also converted to the ATPs. So basically what we are going to discuss today, it is the third step of the cellular respiration, and that is the Krebs cycle. My dear students, the acetyl coenzyme A, first of all, it reacts with a molecule which is called as the precursor of the Krebs cycle, and that is the oxaloacetate. It is something like this. It is oxaloacetate. Basically, this structure is the structure of oxaloacetic acid. But because oxaloacetic acid at the normal biological pH, it is found in the ionized form. So that's why we will write it down as oxaloacetate. So this acetyl coenzyme A reacts with precursor of the Krebs cycle, that is oxaloacetate, and they are converted into the first product of this cycle, that is the citric acid or citrate. So this one is the first product of this cycle, which is called as citric acid, or because of its ionization at the normal biological pH, it is also called as the citrate. My dear students, this reaction, which is basically the step number 1 of this Krebs cycle, it is catalyzed by an enzyme which is called as citrate synthase. And my dear students, in this step, the coenzyme A, SH, that is removed. And what type of reaction it is? Keep in mind that it is an aldol condensation reaction. So this citrate is the first product of this cycle. That is why it is also called as the citric acid cycle. And you can see it is a tricarboxylic acid. So that is why this cycle is also called as the tricarboxylic acid cycle or TCA cycle. Now in the second step, this citrate will be dehydrated. The OH is removed from this carbon and hydrogen is removed from this alpha carbon and water is removed, and now it will be converted to the next molecule, which is basically cis aconitate. And my dear students, because in this step the water is removed from the citrate, so that is why this reaction will be a dehydration reaction. And it will be catalyzed by an enzyme which is called as aconitase. In the next step, now my dear students, the hydration of this is aconitate takes place, and when hydration takes place, the OH of water moves to this CH and the HF of the water will attach to this carbon. And now we will get the isomer of the citric acid, and that is isocitrate. And dear students, water is being added, so this step will be hydration, and it will be catalyzed by that same enzyme aconitase. Now this isocitrate, that is that undergoes the oxidative decarboxylation, means NAD positive, that will be changed to NADH plus H positive. The carbon dioxide will be removed. And my dear students, now this CO2 is removed, and you will get C double bond OH, C double bond O, CH2, CH2, and C double OH. Listen very carefully, my dear students. What happens? This CO2 is removed, and these two hydrogens, they are removed. These two hydrogen, they reduce the NAD, and this one is the CO2 which is removed. So this carbon has now two hydrogen atoms that will be changed to CH2, and because of removal of these hydrogens, the double bond will be formed between carbon and oxygen, and now the product, that is alpha keto glutarate. And my dear students, what type of reaction it is? It is basically oxidative decarboxylation. And as it is oxidative decarboxylation, what kind of enzyme is involved here? So the enzyme will be isocitrate dehydrogenase. The enzyme will be called as the isocitrate dehydrogenase. And now isocitrate was a 6 carbon compound. It is changed to alpha ketoglutarate, which is a 5 carbon compound. Now, my dear students, in the next step, this alpha ketoglutarate, that will again be decarboxylated, and it will be changed into C double bond OH, CH2, CH2, C double bond OH. The structure remains same up till this, and then S coenzyme A. Now, my dear student, this structure is called as succinyl coenzyme A. And again, in this reaction, again what happens? The NAD, that will be changed to NADH NADH plus H positive, mean the oxidation takes place. And my dear students, again this CO2 is removed, so you will say that the CO2 is removed in this step. It is again the oxidative decarboxylation, just like the previous step. And my dear students, the enzyme involved in this reaction, that will also be a dehydrogenase, but now it will be called as the alpha ketoglutarate dehydrogenase. And again, in this reaction, the acetyl coenzyme A, or sorry, coenzyme A, that gets involved. So, my dear students, what happens? This alpha ketoglutarate undergoes oxidative decarboxylation in the presence of the enzyme, that is alpha ketoglutarate dehydrogenase. This CO2 is removed, and this hydrogen, along with the hydrogen of this coenzyme A, both these hydrogen, they will reduce NAD to NADH plus H positive, and this coenzyme A gets attached to this carbon, and you will get a compound, that is the succinyl coenzyme A. Now, my dear students, this succinyl coenzyme A is then changed to succinic acid. This is the succinic acid, because it is again at the in the form of ionized form. It is present in ionized form, so it is called as the succinate. And now, my dear students, in this step, there are just two things that happen. First, the coenzyme A is removed from this carbon, and secondly, my dear students, one ADP joins with the inorganic phosphate, and it is converted to an ATP. So that is the step, the only step where ATP is produced in case of the Krebs cycle, and this succinyl coenzyme A, that is changed to the succinic acid, or you can say succinate. Now, my dear students, this reaction is again catalyzed by an enzyme which is called as succinyl coenzyme A synthetase. The enzyme is called as the succinyl coenzyme A synthetase. Now, this succinic acid that is dehydrogenated and it is changed to the next acid, that is basically fumarate or fumaric acid. And my dear students, it is very easy to understand that in this step basically these two hydrogens, they have lost, these two carbons, sorry, they have lost one hydrogen each, and both these hydrogens, they will be used to convert FAD to FADH2. And because the succinate is dehydrogenated, so the enzyme involved in this step, that will be succinic dehydrogenase. And after dehydrogenation or oxidation, this succinate is changed to the fumarate. Now, my dear students, in the next step, the fumarate, that is changed to malate, and dear students, this in this step again the water is added, and that will be the hydration. And my dear students, now you will get this compound, and that is basically malate. And this reaction is again catalyzed by an enzyme that will be called as the fumarase. So fumarase converts the fumarate to malate, and my dear students, finally this malate will be again changed to the oxaloacetate by the dehydrogenation, and again NAD positive, that will be changed to NADH plus H positive. And that is again a dehydrogenation reaction, or you can say that is basically an oxidation reaction. In this reaction, these two hydrogens are basically removed, and they convert NAD to its reduced form, and this will be catalyzed by an enzyme that will be called as the malate dehydrogenase. So how many steps are there? Total step. The first one is aldol condensation, in which acetyl coenzyme A reacts with oxaloacetate to form citrate. In the second step, this was the first one. In the second step, the citrate is changed to cis aconitate by the dehydration. Now in the third step, again the hydration takes place, and this cis aconitate is changed to isocitrate. Then, my dear students, in this fourth step, oxidative decarboxylation of isocitrate takes place, and it is changed to alpha ketoglutarate. In the fifth step, the alpha ketoglutarate is changed to the succinyl coenzyme A, and again oxidative decarboxylation takes place. And then in the sixth step, this succinyl coenzyme A is changed to the succinate, and my dear students, in this step the coenzyme A is removed basically. Then in the seventh step, this succinate is changed to fumarate by the dehydrogenation. And then, my dear students, in the eighth step, Fumarate is changed to malate. This one is the eighth step. And my dear student, finally the malate is again changed to the oxaloacetate. Now, how many total enzymes are involved in this Krebs cycle? First one is the citrate synthase. Now, my dear students, the second is the aconitase. These two are basically the same enzyme. So that is why total there will be nine steps in this Krebs cycle, but enzymes involved there will be eight, because the second and third step both are both are catalyzed by the same enzyme. Then next is the isocitrate dehydrogenase. Next is alpha ketoglutarate dehydrogenase. The next enzyme is succinyl coenzyme synthetase. The next enzyme, and that is basically succinic dehydrogenase. And then, my dear students, there is fumarase. After fumarase, there is malate dehydrogenase. So all these enzymes are involved, and this is the Krebs cycle. Now, my dear students, there is a very important point to remember, that you will have to memorize the structures which are involved in this Krebs cycle, and it is not a very big task. These are very simple. If you will draw them two or three times, then you will be able to draw all these structures which are involved in this Krebs cycle. Now let us see that how many ATPs can be produced. The one ATP is produced. There is one ATP. And there are, my dear students, total how many NADH are produced? First, then there was a step in which second NADH was produced, and that is this one. And then, my dear student, third. First, second, and third. There are three NADH which are produced, and there is one FADH2, it is produced. Listen very carefully. When one glucose is changed to pyruvate, then the one glucose forms two pyruvate after glycolysis. It means one pyruvate will produce one acetyl coenzyme A, and the second pyruvate will produce the second acetyl coenzyme A. So one Krebs cycle utilizes only one acetyl coenzyme A, and then the second round of the Krebs cycle, that will utilize another acetyl coenzyme A. It means that the one glucose produces two pyruvate produce two acetyl coenzyme A, so there should be two turns of the Krebs cycle to utilize the two acetyl coenzyme A molecules. In one turn, one ATP is produced, so in two turns, two ATPs will be produced. Similarly, my dear students, in one turn there is 3 NADH produced, so in two turns 6 NADH will be produced. In one turn of the Krebs cycle, there is one FADH2 produced, so in two turns there should be two FADH2 molecules that should be produced. So in this way, the two turns of the Krebs cycle produce two ATPs, 6 NADH, and two FADH2 molecules. Now these 6 NADH, when they will enter the electron transport chain, they will produce, my dear students, 18 ATPs, and these two FADH, they will produce 4 ATPs, and these are 2 ATPs. So 2 plus 18 plus 4, that is 24. And my dear students, when the glycolysis takes place, you know that the net gain of the ATPs, that is 2 ATP, it will become 26. And similarly, in case of the glycolysis, there are two NADH which are produced. So 2 NADH, they will again produce 6 ATPs. So you will get almost 32 ATPs when the glucose molecule is oxidized. This is an average calculation, just so one glucose, when it is completely oxidized and metabolized, you will get 32 ATP molecules. I hope so that this lecture will be quite understandable for you people. Next time we will come up with some new metabolic pathway. Till then, take care.